Hello, good evening, everyone. Good evening, Akshat. How are you, Akshat? Good evening, Aryan. How are you all? So, is there any new student today who didn't join yesterday's session? Okay, so let me share my screen. Good evening, Dr. Siddesh. Is the screen visible to everyone? Is audio and video clear? Okay, thank you, Anand. Thank you, Sideshna. So yesterday we talked about what is gene expression. So I hope you understand now what is gene expression. And if there is new student who didn't join the yesterday session, so kindly watch the recording of the yesterday session so it will be much clear. Okay. So yesterday I gave you one situation to imagine where you are working as a bioinformatician and your senior gives you a file, and that file contains a table of 30,000 genes with their gene expression values, right? So, we usually call this table as a gene expression dataset. What do we call it? We call it gene expression dataset. Or simply, we can also call it a dataset. So today, let's talk about what is a dataset, because dataset is one of the most important words in bioinformatics. So let's forget about biology for just 5 minutes and just imagine that your college teacher has marks for 4 5 students. Okay, so let me write here. So just imagine that your teacher has, let's make a table here for student, and we have student like, we have A, we have B, we have C and D. Okay, these here, they represent students. And here, for example, we have marks. For example, any subject, maths or biology, we have marks. So student A has marks 80, student B has marks 75, and here student C has marks again 80 and student D has marks 45. Okay, so now what is this? This is a table, right? This is a table which contains information. So we call this table a data set. Okay, so what is a data set? A data set is simply a collection of related data which is organized into rows and columns. So now let's come back to biology. Now instead of students, imagine that we have genes. So here we have genes and here we have all the gene expression values. So this is also a data set. The only difference is that now the information is about genes instead of students. So a simple rule is that whenever you see a table like this, where you have something here and here you have values of it, here we have something and here we have values of it. So this is a data set. You can always think of it as a data set. So it does not matter what it contains, whether it contains students, whether it contains marks, whether it contains genes or patients or DNA sequences, it is still a data set. Now, why is this important? Why we are talking about data sets so much? So now that we are getting closer to R, so again imagine that someone gives you a data set with 30,000 genes. Now, can R analyze it? So obviously R can analyze it, but first R has to read the data set. Before analyzing it, R has to read this data set. So before learning the R commands, you need to understand that what R is reading, because R doesn't magically know about biology. R does not know anything about biology. It only sees data which is arranged in rows and columns. So here I have one question for you. So here just see this table. Just imagine that I am giving you this table. Now you answer me that what do you think this row here represents? So let me highlight this row. Okay, so I am talking about this row. So what do you think that this row here represent? What information do you think that the single row is trying to tell us? Let me know, what do you think this row is trying to tell us? What information do you think it is giving us? Not the whole table, just this first row. Okay, well received, Dr. Siddesh. Very good, Deeba. Okay, very good, Parth. What about others? Yes, very good, Aryan. Yes, very nice answer, Sudeshna. Okay, so very good everyone. So the answer is, all of your answers are correct. So I will tell the answer once again. So here, if we look at this row, only at this row here, so we can see that TP53 gene expression is more in cancer cells than in healthy cells, right? I can also say it in this way, that the gene expression of TP53 is more in cancer samples compared to the healthy samples. So this means that all of you are thinking about the data. You are not just reading the numbers here, you are thinking about the data, and this is exactly the skill that we want to build. Okay, so the single row here, it is giving us three pieces of information. First, what gene we are talking about. Here we are talking about TP53 gene, and what is it? Its expression in healthy cells, and what is it? Its expression in cancer cells. So from these three pieces of information, we conclude that TP53 has higher expression in cancer cells. Now notice the difference here, that here nothing is written here that TP53 has more expression in cancer cell. Not a single line like this is written here in this entire table. So this row here, it only contains data. We use this data to make a conclusion, and this is how scientists think. So here's something I want you to remember, that there are two different things. First is data, and here we have the data. We have gene, we have expression in healthy cells, and we have expression in cancer cells. So this is the information, and the computer only sees the numbers. As I just said, that there is nothing written here that TP53 has higher expression in cancer. Not any line like this is written here in table. The computer only sees the numbers. And the second thing is interpretation. So we are saying that TP53 is more highly expressed in cancer. So this is our understanding of this data. Now the computer, it only gives us the data, but we are the one who gives it a biological meaning. So this is the actual job of a bioinformatician. So let's understand it one more time with a very simple example. So for example, doctor measures your temperature, right? And the thermometer says that it is 102 degree Fahrenheit. Now, that is data. 102 Fahrenheit, that is data. And the doctor says you have a fever. So that is the interpretation of that data. Now the thermometer does not know what a fever is, right? It only gives you a number. So exactly the same thing happens in bioinformatics. The data set, it only gives numbers. The scientists are the one who interprets those numbers. Now, this is very important in R, or I can say this is why R is important, because one day R might give you a table like this, where I will give you genes, I will give you the numbers in healthy cells, and here I will give you numbers like this in cancer cells. So here R will not say that these are the genes which are causing cancer. R will simply show you that here are the numbers. Then you, as the bioinformatician, you will ask the right questions, that why, for example, MYC is higher here, why PTEN gene has decreased here, or which gene should we study next among all of these genes. So this is why I keep on saying that R is a tool and you are the scientist. So are we clear here with the data set? Okay, very good, Aryan. So now let's talk about how does a computer see this table, okay? Because when you and I look at this table and we think that these are the genes, but computer does not know what a gene is. To a computer, it is simply rows and columns or text or numbers. So once you understand this, R will automatically feel natural, okay? So here, let's see this. So here, suppose that I remove this word gene from here. And let's put question mark here. Okay, so here I remove the word gene and I put question mark here. Now, you tell me that if someone has never studied biology, so do you think that they could tell that this is TP53 is a gene, BRCA1 is a gene, MYC is a gene? If there is a person who has never ever studied biology, do you think that they can tell us that these are all the genes if I remove this word here, gene? Obviously not. They cannot tell that this is a gene. Yes, very good, Himani, very good, Aryan. Nobody can tell that these are the genes if they don't have the knowledge. So, if I show this table to a person who knows biology, so suppose I show this table to a biologist, so he will immediately recognize that these are the genes. But in the same way, if I show this table to the computer. Now, in the same way, the computer does not know biology. To a computer, it looks like that this is the first column which contains text, this is second column which contains numbers, and this is third column which again contains numbers. That's all. So the computer has no idea that this TP53 is a famous gene which is involved in cancer. So this is a very important lesson here, that a computer does not understand biology, it only understands the data. And when you will write the R code, you are not teaching R biology. Okay, you are just simply telling R that read this column, compare these numbers and make me a graph. So you are the one who is providing the biological understanding and R is simply doing the calculation. For example, you use calculators, right? For example, you give two numbers to calculator to add 10 and 25. Now the calculator will give you the answer 35. But the calculator does not know what are those numbers. Those numbers can be gene expression values, those numbers can be exam values, they can be temperature values, or they can be age. So the calculator is, it simply performs the calculation and R works in the same way. Okay, so today here you have reached a very important milestone. You now understand what gene expression is. You now understand what a data set is. You know that a row contains data, and you understand that we interpret the data. We are the one who interprets the data, and you understand that R sees data, it does not see biology. So these concepts here, they may seem very simple, but they are the foundations on which we will build everything else. So are we clear till here? Let me know, are we clear till here? Okay, thank you everyone. Thank you Aryan, thank you Sahan, and thank you Tanushri, Tanu Sharma. Okay, now before we start with the next concept, let's quickly remember the most important sentence that we learned yesterday. So yesterday we learned a very important sentence that R is a tool and you are the scientist. I just repeat that sentence today here as well, that R is a tool and you are the scientist. So today we are going to learn how R sees data. Now notice here that I didn't say how you see data. I say how R sees data. So because this lesson is about thinking like a computer. Okay. Now let's start with how does R see data. So let's start with a very simple question. Okay, so again imagine this table. Okay, we have this table, and when you look at this table, what do you see? You probably see that these are the genes. Healthy and cancer, these are two conditions, and MYC has higher expression in cancer. So you immediately attach biological meaning to this table, right? Now imagine you are a computer. Okay, now the computer does not know biology, it does not know what cancer is, it does not know what a gene is. Instead, it sees something much simpler. Instead of seeing it as genes, computer sees it as column 1. And here, instead of healthy expression, mean expression of healthy cells, it sees it as column 2. And here computer sees this as column 3. So to the computer, the column 1 contains text, the column 2 contains numbers, and column 3 again contains numbers. That's it. Now notice here that everything is arranged in rows and columns, right? Here we have rows and here we have these 3 columns. Now a computer loves this format. Now why computer loves this format? Because it is organized. Can I say that here this data set here, this table here, it is organized? So a computer loves this format. So a computer does not think that this is a gene expression table. It thinks that this is organized as rows and columns, and this is why almost every programming language, not just R, every programming language works so well with tables because they are organized. Now, what does R do? So imagine that you give this table to the R. Okay, you give R this table. Now R will not panic. R will never panic how much huge amount of data set you give it. R will never panic. It simply says that, okay, I have this table, I have these rows, and I have these 3 columns. Now, what does R do? R stores this table into its memory. Okay, R stores the table in the memory so it can answer your questions later. For example, you ask R that show me the TP53 row, or later you ask again question that calculate the average expression in cancer, or later you ask one more question that find me the genes with higher expression in cancer. So R can only do those things because it first stores this table. Okay, so R works with organized data. It does not work with random numbers. It does not work with random words. It works with organized data. And this is why the bioinformatics data set, they are usually arranged as tables. How R can have edge over MS Excel in data analysis. So Dr. Siddesh, we will continue this, we will learn this in our upcoming classes. Okay. Okay. So here in lesson 1, we learned something very important, that R is a tool that helps us analyze the biological data. Is that right? We learned this line from lesson 1, that R is a tool that helps us analyze this biological data. But today we are going to ask a new question, that before R can analyze this data, where does R keep it? Do you all understand the question? Yesterday we learned this, that R can analyze the biological data, but today we are asking a question, that before analyzing this data, where does it keep it? So let's understand it with an example. So imagine that you have just finished an RNA sequence experiment, okay, and your professor emails you a file, and the file name is geneexpression.csv. Okay, you just finished an RNA sequence experiment and your professor emails you a file, and this is the file name. Now, inside this file, there are 20,000 gene expression values. Now, imagine that R opens up this file. So what will happen next after R open up this file? So does R keep reading this file every time you ask it a question? For example, you ask the question that what's the average expression? So does R open this file again? And then later, 2 days later, you ask one more question, that show me the TP53 gene row. So does R open this file again one more time? Then you ask one more question, that draw me a graph. So will R open this file again one more time? So R does not work in this way. It does not open this file again and again, because if R is using this flow of work, so that would be incredibly slow. So R does something much smarter. So imagine that you are working in a laboratory, okay, and your supervisor gives you a box which contains 20,000 records. Okay, you receive this box and it contains 20,000 records of data. Now, would you leave this box in another room and walk back every time you need one record? Tell me, will you do this process, that you will keep this box in another room, and if you need one record, you will just stand up from another room, go to this room, pick up the record, and then go to your room. And then if you need another record, then you will just keep on walking among rooms. You will not follow this path, right? You will not do this. Instead, what you will do, you will just pick up this box and you will keep it on your desk, right? That's the practical way. If you want to work efficiently, what you will do, you will just pick up the box and you will keep it on your desk. So now everything is right in front of you, so you can search it, you can sort it, you can highlight it, you can write notes on it. So everything is in front of you. You don't have to keep walking back to that storage room. So R does exactly the same thing. This CSV file, this is just like that storage room. So when R read this file, what it does, it copies the data into your computer's memory. Okay, are we understanding the point, that this file here, it is just like that storage room? So what R does, it copies the data from this file and it keeps it into your computer's memory. So for example, just like this is your workbench. So in the same way, your computer's memory is just like the R's workbench. So once the data is on the workbench, means in the memory, so R can work with it very quickly. So this is why R does not repeatedly open the file from your disk, okay? It just loads it once and then works with the copy from the memory. So are we clear with this? Did we understand this concept? Okay, very good, Aryan. Very good, Akshat. Okay, we are clear with this concept, right? Okay, but we have one problem here. Okay, we have a very huge problem here. So here you can see this is your workbench, right? And here you have this one box. Now imagine, just like this one box, you have 20 more boxes as well. That means just like this file, geneexpression.csv, you have more files as well. For example, geneexpression1.csv, gene expression 2.csv, gene expression 3.csv. So we have so many files. Now, here we are imagining this workbench, and on this workbench we have several boxes. Now, here, this contains gene expression data. We already know that we only had this single box before and it contains the gene expression data. But now we have another box and that box contains patient information. Now here just imagine that there is one more box. Now we have another box that contains the mutation data. Now we have another box that contains the clinical data. So if none of these boxes had labels on it, your desk would become a chaos, right? If you have similar boxes like this and none of those boxes have labels on it, you will just get confused. Am I right? So what you will do if you don't have labels, you will just constantly ask that which box has the patient data, which box has the mutation data, or where did I put the gene expression data, right? So we never work this way. A scientist would never work like this. So each box gets a label. For example, just here we have 20,000 records written here. So just imagine that we put a label on it that it is gene expression data. So on another box, we mentioned that it is patient data. On another box, we mentioned that it is clinical data, right? So the label doesn't change what's inside the box. Is this point clear that if you put anything on this box, the label will not change what is inside the box. So if this box contains gene expression data and you put the label that it is student marks, so that label will not change the data inside this box. We are right with this point, right? So the label that you put on this, it will just simply tell you where to find it. So R here, it uses exactly the same idea. So in R, these labeled boxes, they are called objects. So this is our first official term, objects. So did we get idea what are objects? So object is simply a place where R stores something. Okay, so an object can store anything. It can store data set, it can store list of genes, or for example, a single number also, or a graph, or anything else. The important idea is that object here, it is just a named container. So are we clear what is an object? Okay, look at here. Look at this image. So just imagine that this is exactly what it looks like inside the R's memory. So here we have that file, geneexpression.csv file. So you can see that that file, it is inside this box, and outside we have the label gene underscore data. Okay, so the name, it is outside and the data, it is inside, just like a label on the box. Okay, now why is this important? Who will tell me why is this important? Why labels are important? Or in official terms, why objects are important? Let me know if you understand the concept of objects. Tanu Sharma, are you still there in the class? Yes, very nice, Dr. Siddesh. What about other students? Very nice, Shobhit. Yes, very nice, Shobhit. Very good, Himani. Very good, Aryan. Organized data. Yes, very good, Sudeshna. Yes, so we all understand about the objects. Very good, Anand, to identify different data sets. Very good, Rashmi, to find information quickly. So, as we now understand that objects, they are very important here. Now, from now on, almost every line of the R code that you will write, it will involve objects. Okay. So, when someone says that create an object, so they simply means that make a labeled container and store something inside it. That's all. So there is no complicated computer science, just an organized way to keep the information. So let's revise what we have learned here before we move on to the next concept. So we understood that your computer has memory, right? And R stores data in the computer's memory, and that data is stored inside the objects, and objects are simply named containers and they do not change anything which is stored inside it. Okay, so for example, if we want to change the name of the object, we can write gene expression data. We can make it, for example, any name that you want, that you would like to give this data set. So this object will not change the data which is present here. Okay, now another question, how does data get into an object? Can anyone tell me that? How does data get into an object? Anyone would like to answer? How does data get into an object? Answer is very simple. Okay, Shobhit, but I would like to see a very simple answer. Okay, so just imagine that you have just starting with your work day and this is your gene expression.csv file and it is on your computer. Okay, Himani, very good. Okay, so here for just imagine that you have started working your day and on your computer, your gene expression.csv file is sitting on your computer. Okay, so at this point, R does not know anything about this file. R has no idea that this file exists. Means this file exists on your computer, but R hasn't opened it yet. Did we get it? That file is there on your computer, but R hasn't opened it yet. R does not know about this file. So R cannot analyze a file it hasn't read. Okay, so suppose you ask R that what is the average expression of all the genes. So R will give you an error. So R will tell average of what? I don't have any data. So R will give you an error. So before R can calculate or compare or plot or filter anything, it must read this file. Okay, and reading means take the information from this file and bring it into the R memory. Okay, so for example, you have a textbook on your shelf. Now, just because you own the book, it does not mean that you know what's written inside it, right? So the information becomes useful only after you open it and read it. So similarly, this CSV file, it is on your computer just like a file. So R here, it must read it before it becomes useful. Now, where does the data go after reading it? So if R reads this file, where does the data go? So again, it brings us back to the object. So when R reads a file, it needs somewhere to put the data. So for example, you are pouring a water from this bottle. Now you need some container to hold that water if you are pouring it from a bottle. So what you do, you use a glass. So simply here, R is using an object. Okay, so the object here, it becomes the container that holds the data set. So are we clear with all the facts that we have learned about today? Do we have any kind of questions related to anything? Okay, so should we see what R looks like? Should we see it now if we don't have any questions? Okay, so here I'm going to open R. So here I'm searching for R. I will also tell you how to download it, but I will tell you later. Okay, so here I have searched for R. So here you can see it is written RStudio. Now, why is this studio here? Because we were just talking about R. So next to R, why studio is written here? So this is something that confuses almost every beginner. So people, what they do, they search on Google, that download R, and then they see another program which is called RStudio, and they wonder that which one are they supposed to download? Should they download R or should they download RStudio? So the answer is both. You need to download R as well, and you need to download RStudio as well. Okay, because they both have different jobs. So I will just explain it with a very nice example. For example, you have a car, and that car has an engine. Now the engine is what makes the car move, right? Without engine, the car will not move. And inside the car, you have a dashboard that lets you to drive it comfortably. So you will not say that the dashboard replaces the engine, right? You need both. You need the engine as well, and you need the dashboard as well. So in the same way, this is a very similar relationship with R and RStudio as well. So R is the engine, means R is the programming language itself. R is the one which is performing calculations. R is the one which is reading files. R is the one which create graph. R is the one that is analyzing the data. So without R, nothing happens. So what is R? It is the programming language itself. Now, this RStudio, this is the dashboard. So RStudio, what is it? It is an application which is built to make working with R much easier. So let's open it. Let's see it. Okay, so this is what RStudio looks like. Is the video clear to everyone? Can you see the RStudio? Okay, so can you see that there are very new things? Maybe you don't understand, maybe you understand, or it might look very advanced to you, or it might look very basic to you, or very common things are also available here. So this is RStudio. It is not R, it is RStudio. So this RStudio, what it does, it provides you a place to write the R code. Okay, this RStudio, it provides you a place where you can write your code. So here it gives you a console where you can run the code. Here it gives you a view of all of your objects. So for example, we understand objects now. So whenever you create an object, you will see all of their view in this area here. And here it also provides you a browsing here, and here it also gives you a help section here. So for example, if you don't understand a thing, or you forgot something, for example a meaning of code, you want to find out how to calculate the arithmetic mean. So here you will just search the topic here, and it will give you all the information here. So here all the information is available how you can calculate the arithmetic mean. So here at the end you also have an example how to write the code to calculate the arithmetic mean. So it also provides you a help section. Okay, and here we have plots window as well. We will also learn about all of these in details. For now I'm just giving you an overview. Okay, so this is RStudio. I have mentioned RStudio as dashboard. I have mentioned RStudio as an application, but in official definition, what is RStudio? So RStudio, it is actually called as an IDE. Has any one of you has ever heard of IDE? IDE means Integrated Development Environment. Has any one of you heard of it? IDE. So what is RStudio in official definition? So RStudio, it is actually an IDE. So IDE means Integrated Development Environment. So RStudio is for R. So in simple terms, R is the programming language that does the actual work, and RStudio is the environment where you run the code, you write the code, you organize it, you manage your R code. Okay, so R is the language and RStudio is the IDE that gives you a convenient place to work with R. Okay, if you don't understand what IDE, so let me give you an example. So for example, you are using a notebook to writing your lesson 1 of R. Okay, and your first notebook is very simple and you have written your lesson 1 of R. Now for your lesson 2 of R, you are using another notebook and that notebook has spirals in it. Okay, so both the notebooks are different, right? Now you have a third notebook where you are writing your R lesson 3, and in that third notebook it does not contain lines. So can I say these three notebooks, they have different features? Notebook 1 is simple, notebook 2 has spirals in it, and notebook 3, it does not contain any lines in it, means it has the blank pages. So can I say these three notebooks are providing different features? So this RStudio is just like a notebook. Okay, so RStudio is not the only IDE where you can write your R programming language. Are we understanding this line? That RStudio here, it is not the only IDE, or I can say it is not the only notebook where you write the R code. So another IDE, for example, we have VS Code. Let me show you VS Code. Okay, so this is we call Visual Studio Code. It is called VS Code in short. So you can also write your R code here as well in VS Code as well. One more IDE is Jupyter Notebook. If you ever heard of it, Jupyter Notebook, you can also write your R code in Jupyter Notebook as well. So what are these? RStudio, VS Code, Jupyter Notebook, what are these? These are the different IDE, means these are the different notebooks where you can write your R language. Okay, but RStudio is the most popular IDE for R, so that is why most bioinformaticians, they work with RStudio. Okay, do we understand this part about IDE? Do we understand now what IDE is? Integrated Development Environment. Okay, so I have one question for you. Can I use R without this RStudio? Yes or no? Can I use R without this RStudio, without any type of IDE? Okay, so if I say only the answer of Aryan is correct. Anand, Deeba, Himani, Aryan, you first wrote yes, now you have written no. Okay, so if I say the answer is yes, you can use R without any of these IDE. So technically, you can install and you can download the R only and you can use it. But however, almost every bioinformatician, every data scientist, every statistician, they use R through RStudio because it is much more comfortable and productive. So let me show you how can use R without any IDE. So here, let's search for R again. So here, can you see this? R and then we have 4.5.1. So this is the version of the R that I am using right now. Okay, so this 4.5.1 is the version. So can you notice the difference between the logo? So this is the actual R language and this is the RStudio. So let's open this R. So this is R only. Let's open it. Now, can you see the difference between the R only and the RStudio, which is an IDE? So here we can see that here in this R, we can only get a console where we can write our commands. Can you see there are no other section here, just like we saw in RStudio? It just only provides a console to type the code. Okay, so this is R itself, this is not RStudio, and here this is RStudio which we are using to write R. So again, RStudio is an environment that makes working with R much easier. So as we can see, if we create any kind of object, we will see it here, but if I use R, I will not see all my objects. Here I can just only write my code, right? Here we can see in RStudio, it provides us this help section. It provides us this file section where we can browse our files. So there is a lot more in RStudio. It provides us with much more features. And here this is simply R and it is very simple and we don't have much features in it. So that's why we use R through RStudio. So I hope the difference between R and RStudio is clear now. Do we have any questions? Everything is clear? The difference between R and RStudio is clear? Okay, very good, Tanu Sharma. Okay, now let me show you how we can download and install R and RStudio both. And from tomorrow, we will write our first line of code and we will learn everything in detail about this RStudio. Okay. Okay, now in order to install R, first of all, we are installing the language. First of all, we are downloading it. Okay, the first step is downloading it. Now, to download the R language, what we have to do, we have to go to the official website. So, the official website, you can just find it by writing R project CRAN. So, if you just write it and click enter, so here you can see this is the official website cran.r-project.org. So this is the official website. So you can just write like this R project CRAN and here you can see the official website. So here we have to go on this official website. Now here we can see download R for Mac OS, download R for Windows. So we will see both of these options to download R for both of the operating system. So first of all, let's see how to download R for Windows. So first of all, you have to click here, download R for Windows. Okay, so this page will appear and what you will have to do, you have to click here on this first line base. As we can see, it is written here, install R for the first time. You can either click on this or you can either click on this. It will just take you to the same page. Okay, so I am clicking here. So here, can we see download R 4.6.1? So this is the version of the R which is for Windows. So what you have to do, you just have to click on this and your R will start to get download. Are we clear how to download R in Windows? It's very simple. Let me just repeat it one more time. So you just have to search for it, R Project CRAN. So this is the official website. You have to click here, and here we can see download R for Windows. Here we can click here on base, or let's click here now, install R for first time. So here we can see the page is the same, and you just have to click here download R, and here you will see in your download section that R has started to download, and you have to download it completely. Okay. Now, if you are using Mac, so you will click here download R for Mac OS. So here you will see these two links here to download the R. So here if you are using Apple Silicon, means M1, M2, M3. So if you are using newer version, you will click here, or if you are using older version of the Mac here, so you will click here if you are using Mac OS. Okay. So after you click here, again you can see that R here has started to download. So we have completed the first step, how to download the R. So once you download it, either you can just directly open here from your downloads, or you can open your file explorer or go to your download section, and you will see something like this. Can we see this? After you download R, you will see something like this. Now, what you have to do now, you have to install it. Downloading alone is not enough. The next step is installing it. So, what you have to do, you just have to click on it. You have to open it, and you just have to click on yes, yes, then next, and then at the end, install. That's it. There are no changes here. You just have to click yes, then next, and then finish setup, install R. Are we clear how to install R? It's very simple. There are no changes in it. You just have to click on next, 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 and then finish setup. That's it. Okay. One more thing. If you successfully install R, what you have to do, let's open RStudio one more time. Just make it a habit that whenever you download or install anything, always check it whether it has properly installed or not. Okay, always check it. So once you install R, so here in this console here, you will type one command, R, it is capital R, okay, dot version. So here we can see r.version.string. So you have to write this command and press enter. So here we can see the version of R. So if you are successfully seeing the version of R, that means your RStudio is successfully using R. But if you see an error, that means your installation was not completed properly. Are we clear with this? After you install R, you have to run this command, and you have to see this version here of R. If you see any error, then let me know in the group, or you can just text me directly. Are we clear with this? Any questions? Okay, very good, Tanu Sharma. Okay, so everyone, that was it for today's lesson. So from tomorrow, we will start using R, and we will learn the R syntax and how to use it in the RStudio. Okay. Sahan, if you are using R in VS Code, then there is no necessity to install RStudio. Just as we have understood what is this RStudio VS Code, they are notebooks. They are just like notebooks. You can use any kind of notebook to write the R language. Okay? You can just move on with VS Code if you want to use VS Code. But here, it's necessary that you learn how to use the RStudio because it provides more features. You can easily and proficiently work with RStudio. Okay. Okay, so if you don't have any other questions, you may leave the class. And for the live students who are watching, so tomorrow we have live class as well at the same time, 7 to 8 p.m. So here we only have 3 sessions which are being offered live on YouTube just as an introduction to this R programming course. So the complete course, if you want to watch the complete course live with me, or even if you want to apply for the recorded lectures as well, so you have to go through the registration process, and the registration link is available in the description. Okay, it is also available in comment as well or in description as well. So if you want to continue with the with this course, so you can register yourself. And tomorrow we have free live class as well. So from after lesson 3, there will be no live classes on YouTube. Okay.